I'd like to introduce Dr. Robert Bode with St. Martin's University in Lacey, Washington. And Dr. Bode will be talking about biocontrol species and how they influence natural selection of broom. So go ahead, sir. And Adam, please turn your camera off.
All right, there we go. Camera's on and good to go. Ooh, there you go. So yeah, thinking about biocontrols and thinking about how they interact with the plant, the first question that often comes up is, well, what if the plant just evolves in response to the biocontrol? So that's really what I've been up to. My last talk in 2017 was about bees, but I answered all the questions I had for now on the bees. So let's talk about seeds and beetles. So I'm looking at the evolution of seed size in these species. So Scotch broom has, well, it's got seeds, and it has small seeds or large seeds, and larger seeds are going to make for larger, more competitive seedlings. And larger seeds may also be able to germinate a little earlier in the season, which gives them a longer growing time, which leads to larger, more competitive plants. So there should be evolution for larger seeds, and that should just kind of go, you know, all the way to, well, why don't plants just have large seeds all the time? And that may be because these large seeds are limited by pressure from seed-eating beetles in the plant's native range, where there's evolution for larger seeds, evolution against those larger seeds, because the beetles are going to be eating the largest of the seeds. So what happens when we introduce this plant and there's no pressure for against large seeds? Well, it could have an increased competitive ability over time. And that's the idea of the evolution of increased competitive ability, essentially that in the absence of natural enemies, there are going to be plants being more competitive. So the natural enemies are here. It looks like I get to be the person to introduce the beetle and the weevil. They're both in the order beetles.
Dr. Bode, just to interrupt you quickly, we don't see slides. Were you intending to show slides?
Oh dear.
Oh dear.
Hey, Ted.
Oh, okay.
Now we see it. Thank you.
Now you
Sorry
see
about
it.
that.
Okay. There's my seeds and such. My apologies. So many buttons. Anyway, the plants want larger seeds. The beetles and other things that are going to eat these seeds are going to want to eat the larger seeds. So you get this balancing selection. That's pretty much where I was. Here are the beetles. You all can see the beetles now, hopefully. We have one over here. It's the mottled seed that you see that's intact but kind of mottled and looks a little almost rotten. That has Bruchidius villosus, which is the Scotch broom seed beetle. And the larvae are going to develop entirely inside a single seed and then emerge in a hole from the bottom of the seed. And the other one you see is Exapion fruscarastre, the Scotch broom seed weevil, which is going to lay its eggs, and the larvae are going to kind of gnaw on the side closest to the placenta, leaving that dent. And they're going to be visible throughout the development, such that when you see the seed, you can see the little weevil kind of moving around in their little weevil larvae. So it's very easy to tell the difference between these two. And I'm going to be focusing almost entirely on the Bruchidius villosus Scotch broom seed beetle, because that one is entirely limited to eating one seed, and they complete their entire life cycle in there. We also know the adults emerge when the seeds are dispersing in the fall or late summer, and that beetles that have laid their eggs on plants with larger seeds are going to have larvae that can grow in larger seeds, which are going to be larvae that become larger adults and have higher fitness. So the fitness of the beetle is directly tied to the fitness of the size of the seed. So Quentin Painter et al., they had this really nice experiment where they saw the evolution of larger seeds in New Zealand, but then the reintroduction of the seed-eating beetles, and are seeing, hypothetically, the balancing again. After reintroducing the natural enemies, they're seeing that beetles are laying eggs in larger seeds, have larger offspring, confirmed it. They're seeing that in sites with higher beetle infestation, there are smaller seeds, as if the evolution has kind of been reversed, that the evolution of increased competitive ability is being pedaled back by the reintroduction of biocontrol, which is very good news. So naturally, I wanted to see how this is going in Washington. We know in Washington that the biocontrol impact is a bit uneven. There are places with a lower impact from Bruchidius villosus. Back in 2017, Nathan Kruger and I published a study showing that at high elevations, there is less biocontrol impact, i.e. fewer seeds being eaten by each biocontrol. So what could be causing this? And we hypothesized that it's the colder temperatures and the higher moisture in the high elevation sites that we looked at. So my questions for this study, the stuff I'm covering today, is really, could there be predictable patterns in biocontrol? And if there are predictable patterns and predictable places where we'll have higher and lower biocontrol, would there be different natural selection in those different places? If there's different natural selection, are we going to see different seed sizes as we rebalance the selection? And if there are different seed sizes, can we use seed size as a proxy for how efficient biocontrol has been over the past couple of years? Now, we can go out and we can measure the presence or absence or quantity of biocontrols and know how efficient biocontrol is for a given year. But natural selection in the past may be visible as seeing smaller and smaller seeds in these populations. So I was wanting to go out and see, do we have smaller seeds in places with higher biocontrol efficiency? And a pattern I was going to use for this was the rain shadow. On the windward side of the Olympic and coastal range, we have lower temperatures, lower average temperatures at least, and higher amounts of moisture. So my prediction was that we would have a lower amount of biocontrol. Plus, this is kind of piggybacking off of what Eric Coombs had found in 2008, where there was lower biocontrol success in moisture, cooler habitats. Whereas on the other side of that red line, in Kitsap, Thurston, Pierce, and Lewis counties, we would expect to see higher biocontrol efficiency, and that would actually be more of a natural selection against big seeds. So we expect to see big seeds on the coastal side, on the windward side, and smaller seeds on the leeward side, and those interior sites. So I did some preliminary study of just 6 sites, just a simple drive up and down, and had my students see the seed destruction rate, how many are being destroyed by beetles and weevils, and what's a seed mass. And that's Sabrina White, who was doing this relatively dull data of weighing tons of seeds. Okay, not literally tons, but large numbers of seeds. And it worked according to plan. It was perfect. Confirmed the pattern we thought we'd see, which was that sites along the windward side of the coastal range are having a lower seed destruction rate, and those same sites have larger seeds. Well, perfect. Well, I learned from Rosemary and Peter Grant, it's always good to get a couple of years of studies in when you're looking at natural selection, because sometimes selective pressures vary. Plus, preliminary study, 6 sites, not really going to be able to publish that. So I came back the next year and I did a whole bunch more sites. These 6 that I'm showing here are the ones that are very close to or were the exact same sites that I used in 2018. So these are 2019 data. I couldn't always use the same sites because sometimes you have a site full of Scotch broom, you come back and it's all gone, you don't mourn, you just find another nearby site, pick up the seeds, see what the biocontrol level is, and see what the seed mass is. And this was a problem. We didn't see the same patterns 2 years in a row. And it wasn't only that we saw a higher level of biocontrol in 2019 than in 2018, we also saw changes in the seed mass, which we would have expected to stay relatively the same between years. Not only that, the seed mass went down in the windward sites, but stayed pretty much the same in the leeward sites, around 8 milligrams. So the seed mass might not be a very valid measurement of the past natural selection, given that seed mass is actually variable between years, even when using the same sites. And site B1 is the same between the 2 years to P1 here. Very different stories. Not only that, we were finding that seed mass might not actually be directly under natural selection. This is Corinne Black down here, and she did an experiment where she was seeing how well the beetles are picking out the biggest seed pods. So you have the length of the seed pod can change, and she measured 10 seed pods for each of these plants and found that on each plant, the beetles will lay the most eggs on the longest seed pods, which happens to be the oldest seed pods. So the beetles will be putting their eggs on the seed pods that are the longest on a per plant basis. Not only that, we started seeing a good correlation between natural selection for, the beetles would lay the most eggs on the longest seed pods on a between plant basis, and also that the beetles would be laying the most eggs on the plants with the largest seed mass on a between plant basis. So here you have one, each line being one plant there for beetles preferring larger seed pods. And down here we have 18 field sites, and each one of those points is going to be the selection gradient for beetles preferring longer pods and beetles preferring larger seeds. These things correlate pretty closely. So our hypothesis now is becoming that seed pod length is what the beetles are actually selecting. Seed mass is just a correlate of seed pod length. We are finding that longer pods mean heavier seeds, so larger seeds. We are finding that longer pods mean more seeds. This isn't exactly novel when it comes to the longer pods mean more seeds. You'd expect you can fit more in them, and Redmond et al. 2000 found that in North Carolina. So kind of just replicating findings there, but it's not at a cost of seed size. So a longer pod has more seeds, and those more seeds are bigger seeds. So if one of these broom beetles is just picking the biggest, longest pod, they're getting more seeds and they're getting the larger seeds, even though the seeds don't finish maturing until the beetles have already finished laying. So the beetles don't even see how big the seeds are anyway. So they're picking the pods and getting the larger seeds. So the natural selection we anticipated is just a subcorrelate of the natural selection that is probably actually occurring, we think. How are we going to find this out? Well, more on that later. So great, I confirmed some findings. Beetles like big seeds, wonderful. Bigger pods fit more seeds, great. Why should anybody actually really care? Well, we're seeing a success story with biocontrol, and that's a bit of good news that we could really use, is that these beetles, these biocontrol beetles, are selectively reducing the fitness of plants with bigger seeds. They are reducing the fitness of our most competitive, most prolific, most fecund Scotch broom. That those Scotch broom we hate the most, they're doing the worst in the presence of the biocontrol. This here is a graph showing that for a standardized seed size in a field site near Centralia, the plants have lower fitness after the beetles have already taken their tolls. We also may be able to use seed size or pod size as a proxy of biocontrol success, but you're probably going to need measurements for a couple different years. And seeing how natural selection has impacted this and seeing actually what the patterns of natural selection are can inform where we may need more biocontrol releases to increase the effects of natural selection. So it could be useful. So the next project is, of course, more field sites. And this is a long-term study. 2020 is going to be year 2 of this. And I'm going to be visiting these 18 field sites, collecting pods, and measuring them so I can determine what the biocontrol amounts are, which is good. Some of the stuff I got from last year, well, measured the pod length, the seed count, how many are eaten by beetles, how many are destroyed by weevils, and what the seed mass is overall on a year-to-year basis to see actually how it does change over time and what the inter-year variation is. I'm happy to say that our 2019 measures, those Kitsap County ones, were very high biocontrol, and it replicates Jennifer Andres' work from 2017, where she had that really nice map of the different biocontrol impacts, our findings are replicating that pretty well. So it looks like it's a pretty reliable one, but there is definitely some inter-year variation, both in the traits and in the levels of natural selection. So we're planning on a 5-year study right now, using a lot of undergraduates to do a lot of counting. And with that, we'll be able to turn all of those little field sites into 5 years worth of data and worth of numbers on how biocontrol varies, how natural selection varies, and how plant traits vary. And now for some fun problems. It could be that there are population spikes of the biocontrol. This is something we will catch in our study, and I think we may have caught one already in Centralia. We saw huge numbers of biocontrol species in Centralia doing a lot of damage and having very heavy natural selection in 2019. And it could be that that spike in biocontrol, well, that's reducing the fitness of those plants, but those plants are perennials. And if next year there is a decrease in the biocontrol, then perhaps next year the plants will have a much higher fitness. So take as a potential effect, we could see a population spike of beetles, followed by a population spike of the wasps that eat those beetles, a population reduction of beetles, and then we would actually have the most fecund year for the Scotch broom. So if we can figure out the patterns of when beetles spike and if there is any pattern in their eruptions, we may actually be able to predict when Scotch broom is going to produce the most seeds and know when to target it so it doesn't. Another thing would be that perhaps the plants are actually responding to the presence of eggs. Now we saw that earlier study where Corinne was measuring the number of eggs per fruit. We went back and we looked at the number of seeds per fruit, and we found that some of the plants just chose not to produce any seeds. They produced the fruit. They didn't produce any seeds in it. And we're wondering right now if it's in response to the bruchins. Bruchins are these compounds in the fluid that the beetle uses to attach its egg to a fruit. Plants can sense bruchins, and plants can start their defenses in the presence of bruchins. So it's possible that a lot of our data is actually being skewed by the plant responding to the presence of a beetle, which it senses when it lays the egg. So it's possible, unfortunately, that Scotch broom may actually be able to evolve a response to defend itself against the beetle. I'm still designing an experiment for this one, hopefully going to get working on that next year. So these are some future directions I may be taking. So some acknowledgements. These are most of the students who have been helping me with this broom research. There's Rebecca Tong, Kaylin Faruda, Corinne Black, and then that picture with 4 people is Maria, Catherine, Julia, and Sabrina. They were all at the Murdoch conference presenting their results. And then on the bottom are Danner and Nathan. Most of these students are on published papers. Corinne, Kaylin, Maria, and Julia are kind of working with me right now on some future stuff. Also, of course, the St. Martin's University, the Murdoch grant number 2015277, and all the people and corporations who funded our nice new science building that I got to use for like 6 months before campus closed. It's got non-carpeted floors in the labs, really high quality stuff there. So I'm very thankful for them. And this is where I normally would be taking questions, but I realize we're waiting till the end. So I'll see y'all at the panel. You can always send me questions or data at rbode@stmartin.edu. I always welcome more questions because they open up wonderful new avenues of research. And with that, I will yield the floor.